I am Lucian Williams in Genetics 311. This is exam 4.2 and today I'll be discussing the spike developmental pathway. While all the cells in your body contain the genetic information to create a whole new you, not every cell ends up becoming the same kind of cell. No matter how trivia night makes me feel, at the end of the day, I'm not just a walking, talking ball of brain tissue. My cells differentiated at early stages of my life and took on different roles. How does this happen, you ask? Through what's known as developmental pathways. Developmental pathways are a series of genes that act to repress or promote the expression of the next gene in the sequence, all accumulating in the presence or absence of some kind of characteristic of a group of cells. Before you're even born, your mother's maternal genes have been transcribed to establish gradients of maternal mRNA in her eggs. This mRNA establishes the axis that you were formed on, as well as control the expression of further genes that divided your newly forming cells into different regions and segments. Identity is given to each segment by homotic genes, giving them color, structure, and function. Through this pathway, we can go from a loose sphere of cells into a structured organism. To explore the function of developmental networks, we will be manipulating the hypothetical spike network, which controls the development of spikes in endoderms of spiky beetles. To clarify what symbols we'll be working with, we have our key here. Arrows will show that a gene promotes the transcription of another gene, while a bar will represent an inhibiting action. Let's begin with a cell whose gene P has been knocked out. Removing P from our sequence means we won't be forming I and can ignore S. Looking at what's left, it seems we have a K being transcribed, which inhibits our spike-forming gene E, so this cell would be smooth. Next, we have an I-negative cell. After removing I, we then disregard our S and P, and are left again with our K gene inhibiting our E expression, so another smooth cell. Next up, we have a double knockout, a cell that's both I-negative and E-negative. We can skip over the I and proceed right down the network to remove the E. Everything upstream is then obsolete, leaving a cell that can't produce spikes, so this too would be a smooth cell. Finally, we have the double knockout of S and K. We can jump right over to K and remove it, as well as everything upstream. What's left is an E gene that's finally allowed to transcribe, so our cell develops spikes. Now, a valid question that may arise from our discussion of the spiked beetle is, why aren't all his ectoderm cells spiky? Well, that's because the developmental network has created regions of the ectoderm where the homotic gene that expresses spikes is promoted and regions where the gene is repressed. In the case of our network, smooth parts of the ectoderm are segments where the transcription of E genes was inhibited by the K gene further up the pathway.